find the original link to this YouTube. Let's go for it.
All right. Well, hello, everyone. Good morning and good afternoon, depending on where you are in the world right now. Welcome to our last live AP Bio review session before Monday's exam. Today is a Q&A session. So we'll be answering some questions that students have submitted through the Google form over the last couple weeks. And then with the time we have left at the end, we'll see if there are some questions that students are asking in the chat. So I'm going to go ahead and share a slide deck. But before I do that, I want to make sure that we here on the panel all introduce ourselves. So my name is Jamie Castle. I teach AP Biology and I'm an AP Bio reader. And I teach at Pennsylvania Leadership Charter School in Westchester, Pennsylvania.
My name is Danielle Taiski, and I am an AP Biology teacher and an AP Biology reader. I teach AP Biology at Trinidad Garza Early College High School and Mountain View College in Dallas, Texas.
Hi, I'm Valerie May. I teach in Woodstock, Connecticut, and I'm an AP Biology teacher and also an AP Bio reader for the exam.
Hi, I'm Valerie Pierce, and I also teach AP Biology at Rutgers Preparatory School in New Jersey, and I'm an AP Bio reader as well.
All right.
I want to jump in there in case kids don't know what a reader is.
Yeah.
A reader is that we spend 8 days in a very large room
Students have submitted
in June grading the AP exams. So we are some of the people who actually read your exam and assign scores to them.
Yes, yes. All right, so I'm going to go ahead and share our slide deck here. Where am I going? All right. Okay, so is everyone seeing the slide deck?
Yep.
On the screen, yes? Okay, great. All right, so a few housekeeping items to get us started. Linked below this video are a number of resources. One of them is the slide deck from the presentation. One is a schedule with links to all of the past AP Bio review sessions. So if you missed the reviews for the different units, we did a review session on labs, we did a review session on FRQ strategies. So if you'd like to go back and watch any of those videos, they will be linked in that schedule. And then there's some additional exam prep resources as well, including the website for the College Board's instructions for accessing the exam. So if you have logistical questions about that, make sure you go to that website. You should all be getting your exam tickets today if you have not already. And if you are not receiving your exam ticket in a timely fashion, then definitely go to that College Board website. I also, just as I'm thinking of it, want to mention that the chat itself is about 20 seconds behind the four of us that are talking. So if you ask questions, chances are the mods will jump on and answer them before us. So, you know, fear not, go ahead and ask your questions in the chat. And even if we don't get to them, the mods will. Okay. So there was one question that I'm going to answer right off the bat in the Google form where questions were submitted, which was, if we watched all the review videos, will we be prepared for the AP Bio exam? And the answer is not entirely. So the review videos are certainly helpful, but I would say that these videos support but do not replace your own review. So, you know, don't count on getting everything that you need from these review videos. A couple of other things to keep in mind, like I said, ask lots of questions in the chat. And AP questions are always going to be about content plus skills. So you're not really going to get questions like, just as a ridiculous example, what is the powerhouse of the cell, right? Or, you know, a little bit less silly, like, what is a property of water? They're not going to ask you questions that directly. They're going to link the content with the scientific practices. And so one of the things that is linked below this video are the AP Biology science practices. And if you're not familiar with them, I would definitely recommend taking a look at those just to be familiar with the types of skills that you may need to demonstrate on this year's exam. All right. Okay. And your panel. We have already introduced ourselves to you. And your moderators, I just wanted to acknowledge, are Lee Ferguson, Maureen Jimenez, and Grace Wheeler. So thank you to them for volunteering their time as well. And we're going to go ahead and get into the questions. So the first question is, what is the role of oxygen in the electron transport chain? So who would like to, from the panel, jump in and take this one?
I will.
Okay.
So you learn it as oxygen is the final electron acceptor. That's the scientific term. What you want to think about is electrons are moving along this electron transport chain, and their energy is being used to do work, to pump hydrogen ions. But as they're passed along, when they get to the end of the chain, they have to go someplace. And that someplace is oxygen. So those electrons go to the oxygen, it grabs more hydrogen ions, and it becomes water, and the water diffuses away. So now there's an empty spot at the end of the electron chain, and electrons can keep flowing. So really the point of oxygen is to remove the electrons so that the flow through that transport chain can continue.
Okay.
Anyone want to add?
I'll just add that from reading a lot of responses on FRQs over the last couple of weeks, one of the things to keep in mind is if there's no electrons, oxygen is not being consumed.
Right.
So if NADH and FADH2 aren't getting their electrons through the electron chain, then oxygen consumption is going to be really, really low. And that's come up in some questions of like, what's going to happen? So level of oxygen consumption if, and then they give you a scenario. And then if there's no oxygen there, there's nothing to take the electrons. And so NADH and FADH2 can't be oxidized.
Yep.
And then that's where you're going to go into the fermentation side of things. Yep. So kind of thinking of those cause effect, because they're going to probably give you some type of disturbance to the system. Like you've memorized that it works this way, and then they're going to throw in a little monkey wrench into it and ask you to predict what happens.
Yeah, especially in that question 2 on this year's exam. There will definitely be a disruption to whatever system they present at the beginning of that question. Yeah. All right. Any more comments, thoughts about our first question here? Oh, Danielle, you're muted.
Okay. I would also like to add that when you look at that inner membrane space, I want you need to think about the fact that, the hydrogen ions are creating an acidic environment, and they are pumped through the ATP synthase to phosphorylate the ADP into ATP. Also think about the name oxidative phosphorylation. You see that the oxygen itself is gaining electrons. That's what makes it oxidative. And the phosphorylation is when ADP, which is the stored form of energy, is converted into ATP, which is a usable form.
Okay. All right. I'm going to go ahead and move on to the next question because I don't think there's any more that I can add here. So the next question is, how is the cell cycle regulated?
Okay. The cell cycle is regulated by cyclins, and those are proteins. And this is another one that could be a good question for number 2, because the disruption could be the denaturation of those cyclins and what happens, which will lead you to cancer. So I can only say for my kids, that's one of the key things that I talk about in heavy detail when it comes to the regulation of the cell cycle. Each one of those checkpoints play a key role in ensuring the viability of a daughter cell. So say, for instance, for the G1 checkpoint, that's making sure that the cell has grown properly. I'm sorry, I can't see. I got nervous a little bit. The G2 checkpoint makes sure that the centrioles, if the animal cell has been produced and is ready to go through mitosis or mitosis or meiosis, and then the M checkpoint makes sure that the chromosomes themselves are properly aligned and or dividing. So that's pretty much how the cell cycle is regulated.
Yeah. I'll jump in and add a little bit too. So not only do we have cyclins playing a role here, we also have these CDKs, or cyclin-dependent kinases. So the cyclin-dependent kinases rely on the presence of certain cyclins. And so what we see here up in this graph in the corner is that the different cyclins are present at certain points in the cell cycle. So we see that the G1 cyclin starts off fairly low in concentration at the beginning of the cell cycle, but then it increases. It increases as we follow the red line and then decreases again. And each of the different cyclins has a different pattern where its concentration increases and decreases. And that's what determines whether these CDKs are active and when they're active at certain points in the cell cycle. So the reason why I threw in this image here was to show how that's working. So the CDK is inactive unless a certain cyclin is present. And that's why you're seeing in this image to the left, the presence of both cyclins and CDKs at certain points around the diagram.
Mm-hmm.
And I'd like to throw in on that theme of a disruption. As you're studying as a student, try to quiz yourself with these questions. Look at the S cyclin. Can you predict what would happen if the S cyclin wasn't made due to some disruption at the correct time? Or what would happen if the S cyclin didn't go away at the correct time? What if it persisted? Can you predict what would happen? That would be a good test for yourself.
Definitely.
I would just add in if there was an inhibitor to the CDK so that it couldn't bind with the cyclin, what would be the effect on progression through the cell cycle? And remembering that kinase is something that adds phosphates. And when those to other things, so there's this kind of chain reaction that's happening. The CDK levels are constant in the cell, but they're inactive when there's not a lot of cyclin. When the appropriate cyclin is there, they become active and they start putting phosphates onto other proteins that then cause other things that make the cell go to the next stage. So anything that gets in there and stops one of those steps from happening would prevent a cell from dividing. But then also, if you had a CDK that was active all the time, even when there was no cyclin present, then you might have a cell that's going through the cell cycle when it's not supposed to, leading to that uncontrolled cell division.
Yeah, lots of potential here for disruptions in systems in this particular situation. All right, moving on to the next question. How do viruses replicate? So I don't know if this is necessarily part of the curriculum anymore, right? It's not. So you don't need to worry about the lytic and lysogenic cycles as you may have had to worry about in past AP Bio exams. But one thing that is mentioned is retroviruses. And
Yes.
I'll admit that retroviruses are not my biggest area of expertise. So if someone else on the panel is more comfortable explaining how the retroviruses work over here on the right, please, you're welcome to do that.
I would just say that retroviruses have RNA as their genetic information instead of DNA.
Mhm.
And so retroviruses are specifically those ones that have that enzyme reverse transcriptase. They bring it with it. It's packaged into the virus particle. They bring their own enzymes with them. And because it's not something our cells do. Our cells don't typically make RNA into DNA. But when we have this retrovirus in us, that RNA from the virus will get copied into DNA. Sometimes that DNA will also get another strand to it, and it actually gets incorporated into the genome of the cell. So with HIV, that's how someone can be HIV positive for a very long time, but their T cells aren't being destroyed by the replication of the virus. When it's time to use the instructions in the virus particle, it could go directly from RNA to proteins, but very often it goes RNA, DNA by reverse transcriptase, and then DNA to RNA by transcription, RNA to proteins by translation to make the new viruses. So they're just kind of a twist on that central dogma that we typically think of as DNA, RNA, protein trait.
Yeah.
If I may, if I may add, when you look at the reverse transcriptase and as well as the process by which it creates DNA from RNA, it also provides a segue into mutations where the HIV virus, and also now with the COVID, where they're saying COVID is a retrovirus as well, you see that they can change very rapidly in their replication cycles. So you have when people take the HIV cocktail itself is trying to break down the parts or the structures within the virus so that it will not be able to attach to the host cell's receptors. So when they look at that holistically, I don't know how they're going to put this in a question on number 1 and number 4, but I know that for the viral replication, or the viral replication itself and how it replicates itself, how it goes through RNA to DNA to RNA, I know that one of the segues that I talk about is how it can evolve very quickly.
My guess is that if they had a question about viruses on this year's test, they would provide a great deal of background information. They wouldn't expect the students to know a lot of detail. They would provide information for you to just apply what you've learned, especially in Unit 6, to that viral system.
I would agree with that with any of the biology concepts. There's going to be a lot of information in that prompt. And reading through that information and understanding what the experiment is and what it's trying to do is going to be really important before you move on to the questions. So jumping right to the questions is not going to be in your best interest. You really can't answer those questions, really for the most part, if you haven't read the information up above. And I kind of been thinking about kind of when to bring your biology. Very often students will say in FRQs, you could have just answered that without learning any biology at all. Well, I think those of us who are readers know that the students who don't bring their biology to the responses don't get very good scores. So it's more like you need to know where to put the biology in. So looking at FRQ number 1, the first couple, like A and B, is bring your biology. What's the biology that's being talked about in this question? If it's saying, how is there a different amino acid there? You say, because there's been a change in the DNA sequence that codes for that protein. Don't just say there's a mutation. Because what is a mutation? Bring your biology to A and B. Yeah. And then the next several parts are going to be really experimental design, positive and negative control, and then the analysis of the data. And then bring the biology back again with another twist to the experiment. So make sure that you read those prompts carefully so you're not answering something about a different experiment than the one that they're presenting to you at the end.
Yeah.
So it seems like there's a very definite pattern, and you got to answer the question that's presented to you, not the question that you think they might want to ask later or something like that.
The context verbs, those verbs are essential. When they ask you to describe and explain, do what they ask you to do. When they ask you to identify, you can answer that in one word, and they're giving points for that, for the identify. And from last year's AP reading, we had an identify question on one of my questions. We had an identify, and they just had to answer it with, just what they had to identify. And it was just a point given. So make sure that you look at those verbs and answer the questions from the verbs.
Definitely. I just want to also point out, because I'm seeing a lot of questions about the structure of the exam coming through in the chat. If you have not taken a look at the videos that College Board has been releasing on the Advanced Placement YouTube channel, the last 4 videos that were produced for AP Biology were practice exams and then reviews of those questions. And those practice exams were structured to be at least as closely to what we expect this year's AP exams to be like as possible. So if you want to get a feel for how many questions are in each of the 2 large FRQs, how many parts of the questions are related to experimental design versus which are related more directly to biology concepts, and the order that they appear in, like Valerie May was referring to before, check out those videos because that's the closest estimate that we have to as far as what you'll expect to see on Monday. Okay. All right.
While you're switching slides, I'm going to just throw something in about explain
Yeah.
and describe.
Sure.
When you see an explain or describe is like, what is it? Like you're answering the question,
Yeah.
what is it? What does it look like? What's the mechanism? What's the chain of events that led to this outcome? Those kind of things. Describe the effect. You're going with increase, decrease, no effect. And then describe this had no effect on this, or this caused this to increase. Explain, you're going to write more than one sentence for an explain. Just kind of think to yourself that one sentence is probably not sufficient for an explain. Explain is always explain why or explain how. So you're getting a little deeper into a mechanism or deeper into reasoning, and you're bringing evidence, that kind of thing. So a very short sentence for a describe or explain is probably not going to be sufficient to earn a point.
Definitely. Yeah. In the resources that are linked below the video, I have a link to a document that I created for my students that has a bunch of other resources in it. And one of the resources in that document is a list of the verbs that you can expect to see in AP Biology questions and explanations of what each of those verbs are asking you to do. So take a look at that if you have not seen that type of a document before. Also, Lee Ferguson did an FRQ strategies review, I think it was 2 weeks ago, and she walked through a lot of this same information as well. So that may be a very helpful video for you to watch as well. Something that I noticed as I was reading exam responses last year and as I read responses from my students in my own class is that often when they don't answer a question correctly, it's not that they got the biology wrong, it's that they didn't answer the question that was being asked. So whatever they write is accurate. They know their biology, but they're just answering the wrong question. So it's so important to understand exactly what those verbs mean within each of the questions. Okay, well, the next question was about the means of horizontal gene transfer. So there's 3 main ways that prokaryotes will transfer genes to one another. We know that prokaryotes are asexual, so they do not experience the crossing over that we see in organisms that undergo gamete production or in organisms that undergo, or the genetic diversity that we see in organisms that undergo sexual reproduction. And so in order for them to have a certain amount of genetic diversity, there have to be other means for them to achieve that. And these 3 are the main means by which they do that. The first one is bacterial transformation. Now, some of you may have done a bacterial transformation lab this year in class, the PGLOW lab. Some of you may have not, depending on where you were in your schedule when school closed. I know my class transformed bacteria literally the day before we closed. So we made it just under the line with that one. But so you'll know if you did that lab that sometimes bacterial transformation can be directed by human intervention, and it involves the addition of a plasmid to a solution into which bacteria are added, and then the bacteria will pick up genetic information from the plasmid. It can also happen, as we're seeing here, that a nearby bacteria may be lyses and releases segments of DNA, and those segments of DNA are then picked up by a recipient cell through transformation. So in short, transformation is the taking up of genetic material from the surrounding environment. Transduction is virus-facilitated gene transfer. So a virus will, a phage, specifically a bacteriophage, will infect a host cell. It will, in addition to adding some of its own DNA to the cell, it may pick up some of the genome of the host cell. And then when more viruses are made and they move on to infect other host cells, they will add not only the viral genome, the DNA or RNA, but also the piece of the host cell's genome from the previous host, and that adds to genetic diversity. And then finally, conjugation is the direct transfer of genetic material through what's called a mating bridge. So two nearby bacteria may form a mating bridge, and then a plasmid, again, this small circular piece of DNA, can be passed from one bacteria to another. Does anyone want to add anything to that?
That was pretty much it.
Okay. All right. Okay. All right. So we'll move on to the next question. All right. There was a question about just basically eukaryotic gene expression overall. So I broke it into transcription, post-transcriptional modification, and translation. So here we've got to focus on transcription and how that happens.
I'll just start with something really, really simple that could be a stumbling block. I've read students saying that the DNA is made into RNA. That's not accurate biology. The DNA is not being made into RNA. The DNA is left intact when transcription is complete. So making sure that you're saying that the DNA is copied into complementary RNA sequence would really hit a lot of the points and show you understand what's going on. And that RNA polymerase is the enzyme that facilitates that process.
I would like to add that something that I have seen quite often is that, you know, at the end of transcription, students have said that you produce a messenger RNA strand that is ready to go out to translation, to go start translation. And in between that, that is a form of gene expression, which is RNA editing that occurs so that when the end product of transcription is a messenger RNA, a pre-messenger RNA transcript that is then cut by spliceosomes, and you have two different types of material that's on there. You have introns and you have exons. Introns do not code for amino acids and exons do. So at the end of RNA editing, you have exons that are bonded together, and you have a 5' cap and a poly A tail that initiates as well as ends translation. And that's when it's ready to go on to translation. And you also have alternate RNA editing that takes place as well.
Yeah. Yeah, so this image illustrates what Danielle was just talking about the post-transcriptional modification that happens, the addition of the cap and the tail, and then, yeah, this alternate splicing that can take place. Yeah. So we'll come back to that. I just want to just hit this really quickly. So this image does a really good job of explaining what happens in each of the three main phases of transcription. The first one would be initiation. That's when the RNA polymerase actually attaches to the DNA template. And you can read in the slides, you know, the longer description here. The elongation phase is where the RNA polymerase is actually synthesizing the messenger RNA transcript based on reading the DNA template. And then finally, termination is when the RNA polymerase detaches from the DNA and the mRNA goes into that post-transcriptional modification that Danielle was just talking about. Something I do want to point out as well is that you can see that the RNA polymerase produces the mRNA molecule in a 5-to-3 direction, which means that the RNA polymerase is reading the template DNA in the 3' to 5' direction. And that's an important distinction to understand. I also know that it's a sticking point for a lot of students remembering which is which. So just make sure that you spend a little bit of time really solidifying that in your mind. I agree with what Val was saying about students sometimes struggling with the words to explain what is happening in transcription. Sometimes they'll just say that DNA is copied, and that is not sufficient because DNA is copied during DNA replication, but right. We're not making another DNA strand here. We're making an mRNA transcript. So if you're going to use the word copy, it's really important to explain that the copy that's being made is an mRNA transcript that is complementary to the DNA template. It's not a copy of DNA. Yeah. All right.
One thing easy to remember, DNA polymerase does this and RNA polymerase does this. they can only add to the 3' end of what they're making. If
Right.
you remember that one phrase, you can kind of then work around, like which way is it reading, which way is it synthesizing, all that type of thing. So just remembering RNA polymerase, DNA polymerase can only add to the 3' end of the strand that it is making. I mean, it's because there's a little hydroxyl group there, but dehydration synthesis has to happen. So you're going all the way back to unit 1. You need that hydroxyl group to come off so you can connect the monomer. But don't worry about like memorizing every tiny little detail. Kind of have a starting point, and then you can move through the logic.
Right.
And I'd like to add, I see sometimes students get confused about this compared to replication because the RNA polymerase only is making one strand. It can just make the new strand 5 to 3 in its preferred direction. You don't need to have Okazaki fragments. You don't need any ligase. You don't have that lagging strand problem in transcription that you do in replication. So sometimes, so just realize that there's only one strand. It picks the easy one because who wouldn't?
Yeah, that's a great point for sure. All right, so Danielle already went into post-transcriptional modification, but then she started talking about alternative splicing. So let's talk a little bit about what goes on there. Does anyone want to explain that a little bit?
It's this weird thing that eukaryotes do. It's the explanation for the finding that just didn't make sense. We found that in many eukaryotic cells, there's way more proteins than there are genes.
Right.
And so it had always been kind of thought, one gene, one protein, that's it. But then there's all these different proteins, and we find that there's not that many genes in the genome. So then they discovered that there's these groups of related proteins, and it becomes from the same gene copied into the same primary transcript can be spliced in different ways. So the gene is, nothing happens to the gene. The gene doesn't get spliced.
Right.
The primary transcript, because of signals inside the cell that is way beyond the scope, like the mechanism of alternative splicing is way beyond the scope of what you need to understand. Understanding that it can happen, that's important. And so not all the introns actually get kept as introns. Some of them actually get spliced out. And it helps to explain something that was a paradigm that was causing problems in figuring out coding for the genome.
And I tie it back to that big idea one of evolution, that evolution can't just create new things out of thin air. It builds on what exists.
Right.
So they had these genes and they could build and diversify from them by shuffling the pieces.
Yeah.
I think
oh, go ahead.
I think one of the big things that kids need to take away from is the fact that pretty much everything goes back to evolution. When you look at the overarching, idea of, biology, it goes back, everything goes back to evolution. So if you can look at this and say, and, like she said before, you don't have to know the processes of alternative splicing, but just know the idea. And you could tie that back to evolution if they have a question like that. Now, I don't know if they're going to have a question like that, but if they do, that would be beneficial for you.
Yeah, they most likely won't have anything directly related to natural selection because I know that unit is not included in part of the testing, but there is a topic in, I believe it's unit 4 or unit 5 about fitness of organisms. Yes. So don't assume that there'll be nothing that sounds anything like evolution on the exam.
Yeah.
There will be a little bit on fitness. Go ahead.
And unit 6 also has some information about the idea of the effect of a mutation. So just keep in mind that mutations aren't always bad. They're not always good. A mutation is just a change in the DNA sequence. The effect of the mutation is based on how does it affect the protein that is made? Does it affect its shape? Does it affect if it's made? Does it affect its shape? If it affects those two things, then it might affect the function, and then that might affect a phenotype, and then that is what is selected for or against. That's where the benefit or the negative effect of a mutation comes into play. Is how does that mutation affect the phenotype of the organism? And that's explicitly shown in Unit 6. So I can see that as fair game.
Yeah. Okay, so just to come back to alternative splicing and show you quickly what it looks like. So let's say our exons are numbered 1, 2, and 3. And the idea of alternative splicing basically says that we can make a number of proteins from one mRNA transcript. So maybe this protein is, 1, 2, and 3. All 3 exons are included. But there's also a possibility to make a protein out of just 1 and 2, or just 2 and 3, or just 1 and 3. And so there are a lot of different options available, even just in this example mRNA transcript with just 3 exons. So the idea that you can get more than one protein from a single mRNA transcript is really the kernel of the idea here that is most important to retain. Okay, let's move on to translation. So translation also has the same 3 phases that we saw in transcription. So there's an initiation phase, an elongation phase, and a termination phase. The big difference here is that translation is happening outside the nucleus. So transcription is happening inside the nucleus where the DNA is located. But then once that mRNA becomes processed, it can move through a nuclear pore out into the cytosol, and it will join up with a ribosome. And that's where the translation happens. As the mRNA molecule moves through the ribosome, it joins together with these tRNA molecules that move into these E, P, and A sites. And then these tRNA molecules have amino acids attached to them. And these tRNA molecules also have what are called anticodons. And so the tRNA molecules will come in here and attempt to match up with the codons on the mRNA transcript. When these tRNA molecules match, then the amino acid from that tRNA molecule will be added to the growing amino acid chain. What else? What am I forgetting?
The anticodon does not code
Thank
for
you.
the amino acid. Okay.
Right.
So when you guys are looking at this, and I am a stickler on this with my students, the anticodon is not coding for the amino acid. The messenger RNA is the codons on the messenger RNA strand codes for the specific amino acid. Okay. The amino acids are, I can't do this, but the amino acids are coded from the messenger RNA. Please keep that in your brain. Okay, keep that in your short term right here. I need you guys to keep that in there, as well as, you can go back to Unit 1 as the binding of the amino acids, they link together by peptide bonds. Okay, so make sure that you guys, remember that, as well as at the end of translation, you have in eukaryotic gene expression, you have a polypeptide sequence, and then it goes through different organelles to be packaged and distributed inside or outside the cell.
And the ribosome is the catalyst making that peptide bond. There's not a single enzyme like RNA polymerase or DNA polymerase. It's the ribosome that's the catalyst in this process. And I remind my students, they do not expect you to memorize which codon corresponds to which amino acid. If they want you to predict a protein sequence, they will give you a table, or you can look it up, but they will not expect you to memorize.
Yeah, and I would add that this is where the effect of substitution, insertion, and deletion mutations in the DNA, this is where the effect will show up. Right. Is in translation, because it's going to change what those codons are. So substitution is just going to change one letter, which may not change the amino acid sequence at all because of that redundancy. And again, if they're going to ask you anything about understanding what the mRNA sequence means in terms of the amino acid sequence, they're going to give you a codon chart to refer to. And And then remember those insertions or deletions of 1 or 2 are going to change that reading frame because the key is 3 letters at a time for a codon. So anything that shifts what those 3 letters are from what they would normally be without the mutation back in the DNA. And that's important to remember. The mutation was back in the DNA, not during transcription, back during replication some time ago. And that's that frame shift mutation that really changes the amino acid sequence a lot.
And just to add to Ms. May, when she said that the trait would be shown or the phenotype will be seen, that protein will express the trait, okay? The protein that is being produced at the end of, well, once in eukaryotic gene regulation, once the polypeptide is being packaged and distributed, that particular protein that's made will express a trait. So that's another tie-in to evolution as well.
Right. Next question is about how histones affect gene expression. So now we're moving out of expression and into gene regulation.
This is another example of gene expression where you have methylation and then you have acetylation. Methylation is basically when the histones bundle up and they become tightly packed. So therefore the transcription factors cannot bind to the DNA. Okay, so that means that no transcription can take place because those histones are tightly wound around that DNA molecule. When acetylation is the opposite, is where the DNA molecule becomes loose. So therefore transcription can take place. So when you think of methylation, just think of inhibiting transcription from taking place. When you think of acetylation, transcription can take place.
So every year I kind of joke with my students that I say something like, if they're going to ask you that level of detail, they're just being mean. They won't do that. And then every once in a while, there's something that comes out where it's yes. So there is this course description. If there's an essential knowledge in the course description, it's fair game. And so knowing that methylation inactivates, it's in there. So one of the things that we came up with my class is that methyl groups are CH3. They're very nonpolar. Nonpolar things like to be together away from water. And so that kind of, this is a total oversimplification of what's going on.
Right.
But it's a good way to remember it, that all those methyl groups are going to make those histones come together, and the DNA is not, all those proteins that are involved in transcription can't get in there and can't transcribe the gene. It's kind of like zipped up tight. Acetyl groups actually have carboxyl groups in them, and they become a negative charge. And so all those negative charges are not going to be near each other, and it's going to cause the histones to kind of spread out. So methyl groups coil it up, acetyl groups loosen it up, and then you go with loosened up can be transcribed. It doesn't have to be. It just can be. It's accessible. So it's that first level of gene expression regulation that we have. And
this ties to cell specialization.
Yep.
Because we know that all somatic cells have the exact same genome in them, but we certainly don't, all those cells don't appear the same or function the same. We would never mistake a muscle cell for a neuron. So one of the key differences in those cells is the methylation and acetylation, where that's occurring in the genome. And that allows these cells to specialize and perform specific functions.
So a question just came up in the chat about whether the DNA is still wrapped around histones, even when it's not. They say arranged into chromosomes. I think they're talking about condensed chromosomes. And we should just think like DNA is always wrapped around the histones.
Right.
It's how tightly coiled up it is after that point will determine this heterochromatin, where it's tightly wrapped, full chromosomes wrapped up for mitosis and meiosis, but DNA is always wrapped around those histones.
Right.
Yeah. Okay. All right. Another big method of gene regulation is gene switches. Now, this is specific to eukaryotes. Gene regulation in prokaryotes is going to happen through operons. So that's an important point to make note of. I love this diagram because I do this activity in my class with my students,
Me too.
and we draw these diagrams out on the table. And so the big takeaway here is you have certain points upstream from the actual coding region of the DNA where transcription factors can bind and either promote or inhibit expression of the gene downstream. So over here on the right side, we see the gene switch here, indicated by the distal control elements. And then we have activators here, here, and here. Those are the transcription factors that are binding to those distal control elements in the gene switch. We also see it over here on the left. These are the switches for the stickleback gene. And depending on where in the body the particular cells are located in the stickleback fish, certain switches will be turned on and off depending on which transcription factors are available and which switches they match up with. So we see in the jaw, the switches match up to the jaw switch. In the pelvis, they match up to the pelvic switch. In the eye, they don't match up to any of the existing switches. But then in the pituitary, they match up to the pituitary switch. So whether or not a gene gets expressed is often a function of the availability of transcription factors that will bind to a switch to promote expression of that gene.
So I'll just jump in that this is another opportunity for mutations to occur. So it's not mutations in that coding sequence for the PIDX1 gene for this example. It would be a mutation that would kind of knock out one of those switches. Take out, either delete that whole section of DNA or change the sequence so that the activator could no longer bind. So in the case of this it's a 3-spine stickleback, a mutation has occurred long time ago, passed down to many fish, where there's a mutation that takes out that pelvic switch. That sequence is gone. And so what happens is these fish don't express the PIDX1 gene in their pelvis. They express it other places because those switches are still there. And they don't make the spines on their pelvis. Now, is that good or is that bad? Well, it all depends. And this is also in Unit 6. What makes it good or bad is the environment that the fish lives in. So if it's not an advantage to have the spines, then that's the advantageous trait. That's the mutation that's going to be beneficial.
And they're not expecting that you will have memorized or know specific examples like this. Again, they will give you the information in the diagram. And as a reader, one of the questions I graded had a diagram on a system and the kids had to answer it. And a lot of students lost points because they were vague in their reference to which elements of the diagram they were writing about. So in this example, for example, if you just wrote the switch is missing, there's 3 switches. We would need you to say which switch is missing for us to give you the point. So use the words in the diagrams they give you. Make sure you use the terms for the same item so the readers know what you're referring to.
Definitely. All right.
And I'll just add to that, if they're asking you to describe population A and population B, then do both of those things separately and actually say, this is what's going to happen in population A. And then another sentence that says, this is what's going to happen in population B, because it might be the same thing happening in both. It might be something different, but that word "and" there is meaning that they want you to specifically address and say population A and then population B. Mm-hmm. Kind of some of the tricks of making it so that you're communicating what they want you to communicate.
Yeah. Okay. There was a question about the role of acetyl-CoA in the citric acid cycle. So acetyl-CoA. How do I want to word this? I'm having difficulty coming up with the right phrasing here. Acetyl-CoA is created from pyruvate. The pyruvate is produced at the end of glycolysis. And then acetyl-CoA goes into the citric acid cycle and is basically the raw material that initiates one rotation or one cycle of the citric acid cycle.
Yeah.
You do not need to know the steps of the citric acid cycle.
Right.
So please do not spend time memorizing those steps or any of the enzymes or any of that. It is important to understand that there is a small amount of ATP production in the citric acid cycle, but that the major benefit of the citric acid cycle is the production or is the movement of electrons onto the electron transport or the electron carriers, so that those electrons can be brought to the electron transport chain. So that's the big benefit of the citric acid cycle, is basically the liberation of electrons from the carbon compound to move on to the electron transport chain, where we get the largest amount of ATP yield.
Right. So to tie back to our previous discussion of the ETC, the acetyl group on that acetyl-CoA is getting oxidized. Its electrons are being pulled off and put onto NADH and FADH2. And when you oxidize carbon compounds, they become carbon dioxide. That's the most oxidized form of carbon. And so this citric acid cycle is oxidizing it, as oxidizing the acetyl-CoA.
Mm-hmm.
Yeah, and I would add that everything that we're actually seeing in this diagram, the purpose is not really to make ATP. The purpose of glycolysis, pyruvate oxidation to get us that acetyl-CoA, and then the citric acid cycle is to get all of those high energy electrons harvested off so that they can go to the electron transport chain and we can do oxidative phosphorylation.
Right.
And when we don't have oxygen available or there's something disrupting that electron transport chain, we can at least keep glycolysis going because we've got that fermentation step that will take the electrons off of NADH without oxygen and give NAD+ back to glycolysis. So another disruption idea to a system that you might have really well memorized, and then, okay, so what happens if such and such enzyme isn't working? What is the cell going to do? And then the eukaryotic cells that have mitochondria, they typically cannot rely on fermentation as a way to make ATP. It is something that can only be done for a very, very short amount of time. So that's sometimes a misconception that kids have, that like, oh, you can just do fermentation instead.
Right.
No.
And I think to highlight, they don't need to know the exact numbers, but aerobic respiration produces much, much more ATP.
Right.
And it gets much more of the energy out of the carbon compounds. And I highlight to my students to think about the idea that carbon dioxide doesn't burn. You can't burn it. Ethanol burns. It still has energy in its bonds. And so that represents that energy that wasn't converted to ATP energy. It wasn't harvested. It wasn't put into ATP. So it's less advantageous to do fermentation if you're capable of respiration.
Right.
And then looping it to the photosynthesis aspect of it, we often focus a lot in photosynthesis about the carbons. It's really, again, all about the electrons. The carbons are just carrying the electrons around. We eat organic compounds to get building blocks, but to get those electrons as well. So in the light reactions, the whole point of the light reactions is to energize those low energy electrons that are in the water molecules to get them to be these high energy electrons that end up in the glucose. So we think of the carbon cycle, but we also really want to think of the energy flow that happens here, starting with light in most cases, going to those high energy electrons in the organic compounds, and then being broken down through cellular respiration to provide us with the energy to make ATP. So kind of making those connections is important. And if you say the word ATP, use the word energy. Never just mention ATP on its own. Put the word energy in there, because that's what it's there for.
Okay. That actually leads nicely into what is the difference between oxidative phosphorylation and the electron transport chain. So the electron transport chain is a term that just refers to the movement of electrons through a number of membrane proteins to essentially drive hydrogen ions across the membrane. Now we see the electron transport chain in oxidative phosphorylation and cellular respiration, but we also see it in the light-dependent reactions in photosynthesis. So when you're thinking of what's happening specifically in cellular respiration, electron transport chain isn't exactly the specific word, right? What you're looking for is oxidative phosphorylation. And then at the end of that chain, then we have the chemiosmosis that drives the production of ATP.
Right.
Yeah. And that's a question that comes up in nearly every year too. What is the difference between the electron transport chain or the oxidative phosphorylation and chemiosmosis? I feel like chemiosmosis is a term that students struggle to understand a little bit.
Yeah, I
You
would
find
say
that
that
as well.
Yeah, I would say that oxidative phosphorylation is the overall process that involves the electron transport chain that makes that proton gradient with that stored energy in the intermembrane space, and then chemiosmosis, which uses that proton gradient to provide the energy to phosphorylate ATP. So I would think the way I think about it is oxidative phosphorylation is the whole thing, and it has two parts, the electron transport chain and chemiosmosis. So then there's photophosphorylation in the light reactions, which also has an electron transport chain and chemiosmosis with ATP synthase. So learn it once, use it twice.
Yeah.
It's pretty, and then know the difference between the two.
Know the difference between the two.
Yep.
Of knowing what is the goal of the light reactions in photosynthesis as opposed to oxidative phosphorylation in the mitochondria.
And think of it as a transformation of energy. So these high energy electrons are coming in that originally came from the oxidation of organic compounds. That's the source of energy. So that's why we call it oxidative phosphorylation. Then those electrons, the energy from the electrons is now transferred into the proton gradient. So there's a lot of potential energy there because those protons can't just come back through the membrane because they're charged. Those electrons now have no energy, so that's why oxygen takes them and goes off and becomes water. And then the energy, that potential energy in the proton gradient, those protons come through and now looping it into unit 3. Which one's cell transport?
Transport is 2.
2, yeah. So now you're looping in some cell transport in here. And so those protons are going down their concentration gradient. And so we have facilitated diffusion happening through that ATP synthase. And that energy is now kinetic energy because it's their flow. And then that energy gets trapped into the ATP molecule. So it's all about the energy and where it is and where it's going. And the energy is completely, totally lost at every step along the way. In the past, there was a question where you had to calculate the efficiency of the process. And then what happened to the rest of the energy? And that energy is lost as heat, keeping us nice and warm.
I often get the question, well, why do cells bother making ATP instead of just using the glucose? And it's that some of the enzymes and proteins that need energy can't access the energy in glucose. Your sodium potassium pump needs it to be in the form of ATP. Muscle contraction needs energy in the form of ATP. That's why you have to go through these processes and lose some of that energy.
Yep, so they're made to have a phosphate added to them. And when that phosphate is transferred to those proteins, it gives those proteins energy to change their shape and then do the work that they need to do.
All right. I knew this was going to come up before I even looked at the questions for today. But there's a question about how to calculate water potential. Now, is this something that they're going to have to worry about on this year's exam? Yes?
Yep. Unit
okay.
2.
Okay. I just wasn't sure just because of the way that the calculator's not indicated as something they may need for the
True.
exam. So I didn't know how
True.
much calculation they really had to do.
Right.
But yeah, it's normally part of the curriculum for sure.
Yeah.
Yeah.
Right.
They have said that all math can be done without a calculator. This math would be a little bit challenging without a calculator,
Mhm.
and time consuming because you're multiplying by that 0.0831.
Mhm.
But I would say understanding the relationship that the formula presents would definitely be something that could be fair game. So really just understanding that because of that negative sign there, as you increase the solute concentration, you decrease the potential that water will leave that solution because it's hanging out with all of these ions. So think of ions that are trapped on one side, the water is gonna form those hydration shells there. So more solute, water moves less. And so when we say high water potential, we mean closer to zero because of that negative right there.
Yeah. I usually advise my students to use more negative or less negative, just because it makes it so much easier to understand than higher or lower, because
Right.
we're talking about negative numbers. Yeah.
Yep.
Yeah.
So because if there was no solutes in the solution at all, C would be zero. So the water potential of distilled water is zero,
Mhm.
the highest water potential due to solute that you can get.
Yeah.
And those water molecules, because there's no solutes trapping them on one side of the membrane, those water molecules are free to move. So they're more likely to move to the other side and then get trapped by those solutes. So that's where that hypertonic and hypotonic come into play.
Right.
So hypertonic with more solutes typically is going to give you a lower water or more negative water potential.
Right. Yeah, I think last year or 2 years ago, there was a question that involved water potential, and students could use the terms, or they could use the terms hypertonic, hypotonic, hyperosmolar, hypoosmolar. As long as they did it correctly, any of those approaches were okay.
Right.
Yep.
Yeah. And this may be because I did the Unit 1 review, but I just kind of want to point out this I variable here is going to depend on whether your solute is ionic or covalent.
Right.
So if you have an ionic solid like sodium chloride, that's going to split up into its constituent ions. So there's going to be 2 particles for each individual particle of sodium chloride that is dissolved in the solution. But if you have a covalent compound like glucose, covalent compounds do not break into pieces in water. So that's a 1-to-1 ratio for something like glucose in water. So that's an important thing to remember relative to these calculations. All right. Okay, so those were all the questions that were sent in ahead of time. I've been looking through, I've been glancing at the chat from time to time as we've been going through, and it looks like the mods are doing an amazing job at answering content-based questions that are coming through that were not related to what we've been answering. But I'm also seeing a lot of questions as far as like, how long are the questions going to be? How many parts are the questions going to be? And my advice for that would be, don't worry so much about how many parts are going to be in the questions. As long as you know what the structure of the questions is going to be like, you will be well prepared. So if you have either talked with your teacher, or if you watch some of those advanced placement practice exam videos, you will see the structure of how question 1 and question 2 will be. Question 1 will give you some visualization of data and provide you with some kind of an experimental setup, and we'll explain and we'll ask you questions about that. There will be questions about both biology and about experimental design. In the second question, it will be a little bit more concept-based, and there, as we've talked about earlier, there will be that disruption in the system at some point. So you'll have to answer questions about the system both before and after the disruption.
Make sure
go ahead. Oh, this is really quick. I've seen this question both in the chat and on Facebook. So when they say that a calculator is not necessary, it doesn't mean it's not allowed. So you are allowed to have a calculator. If you don't trust yourself doing some division in your head or on a piece of scratch paper, you can have that calculator, and it's any calculator you want. So just because it says it's not necessary does not mean it. So it is allowed. A calculator is allowed.
For both questions 1 and 4, when you make a prediction, make sure that you can justify that. Okay, because when you look at number 1 on part D, it says make and justify predictions. Once you make that prediction, make sure that you can provide information that will support your prediction.
Yeah. I'm seeing a lot of questions come up about action potential. You do not need to know action potentials anymore in AP Bio. None of the body systems are required as testable content in the AP Biology exam anymore. It used to be up until last year that endocrine system, nervous system, and immune system, there were aspects of those systems that we studied, particularly as illustrations of cell communication, but those 3 systems have even been removed. So anything related to anatomy and physiology, you will not be required to know that material. Now, that doesn't mean that you won't see a scenario on the exam related to a body system. However, whatever content-based information you would need in order to answer the question using your knowledge of AP Biology will be provided in the question. So no, you do not need to know how an action potential works, but if there happens to be a question about an action potential, they will tell you enough about action potentials that you can then use your AP Biology knowledge to answer the rest of the question.
Mm-hmm.
Yeah. I'm just glancing through here to see what else. Hypo. Yeah, there's a lot of action potential questions that came up. Okay.
Yeah,
Go
with
ahead.
the action potential, the only thing that I could see as being fair game with that is understanding the sodium potassium pump. And that what it's doing is it's actually creating what's called a membrane potential, where there's a difference in the charge outside the membrane versus inside the membrane. And that's only important in that it adds another reason why ions might go in or out of the cell. So that electrochemical gradient concept, that ions will go down their concentration gradient, but if there's a positive ion and the inside of the membrane is positive, even though there's a lot of positive ions outside the cell, it's probably not going to go into the cell because positives will repel from each other. So if they had a question about action potentials, yes, they would give you tons of information, but you would need to understand how that applies to cell transport and how ions would move across the membrane.
Okay. I just got a message from one of our moderators about standard error of the mean. There were some questions about that earlier. So it might be helpful for us to talk about that. So standard error of the mean, let's see, I'm going to unshare my screen for a second and see if I can find a good image that we can use to illustrate that.
While you're looking
while I'm doing that, does someone want to start talking about it?
I'll just give a really quick, short, like what is standard error of the mean? So there's standard deviation and there's standard error of the mean. They are different things. They tell you different things about your data set. So the idea is that you have a sample with a bunch of measurements. Your standard deviation just tells you how spread out those measurements are from the mean. What's the variation in your measurements in your sample? The standard error of the mean gives you an indication that if you were to collect a sample multiple times, if you were to do this same experiment multiple times, if you were to go out and collect a bunch of lizards multiple times and from the same population, from the same group, which is something you would never do because that would be time consuming. Mm-hmm. This is your estimate of where the mean should fall based on your sample size and based on your standard deviation. The way the formula works, which you have on your formula sheet, you don't have to calculate it, but you need to understand it, is that you can make the standard error of the mean of an experiment smaller simply by having a bigger sample size because you're dividing by square root of n. It's giving you some confidence of where do you think the true mean of the population should fall. That's the idea of the standard error of the mean.
I found a good graph, so I'm going to go ahead and share this. Okay. All right, so what we see here, and it doesn't matter exactly what the data is, but what we're seeing are, means and then standard errors of the mean. So the means.
Oh, wait, Jamie, these say standard deviation. Oh, wait,
Do they?
standard deviation. Oh, the first one is showing standard deviations. The second one is showing standard deviation of the mean. So the second one is standard error of the mean.
Okay,
Right.
all right. So we're going to look specifically here at this graph on the right. So what we're looking for in terms of statistical significance of the data is whether or not the error bars overlap. So I've seen a number of questions on the AP exam that ask for, for example, the effect of a certain variable on a certain dependent variable. You have to look at the graph and see where the standard error bars are located. If the bars overlap, then there is no statistically significant difference between the means of the two populations. Okay, so for example, here, the apples and oranges groups are statistically significant from the plums group, but not from one another. So we can see very clearly that the standard error bar for the plums group is much, much lower. But when we look at the two error bars for the apples and oranges group, they do overlap. So that means that there is a possibility that the mean for apples and oranges could be the same within the margins of that standard error. So when you're looking at a question with standard error, that's what you want to look for to determine if there's a difference or not.
And one of the things that I see students say, they kind of get the idea that they're, when they don't overlap, then this is affecting that. When they do overlap, they sometimes just want to say there's no difference. Well, if it's asking you to describe the effect, a difference is not an effect. You would need to take that one step further and say, I'm trying to read it from this graph, like the price, whether you are buying an apple or an orange has no effect on its price or something like that. So just saying that because the error bars are overlapping means that there's no significant difference. You haven't gone far enough to describe what the effect is.
Yeah.
You have to go to say no effect.
Yeah, I apologize for this not being the greatest example
No,
in terms
that's
of
okay.
variables. I saw the bars overlapping and I said, oh, perfect. So
Yeah, no, it works. So if you go with standard deviation with the graph A, so if they tell you that the error bars are standard deviation error bars, not standard error of the mean error bars, all that's going to tell you, what we can say from this graph is that the prices of oranges has a lot of variability in the United States because that error bar is so big. Whereas there seems to be less variability in the price of an apple. And you could say that sometimes an apple costs the same as an orange, but it never costs the same as a plum.
Right.
A plum is always cheaper. And then when you go to B, maybe this is data taken from different markets or different years, or you want to know, does a apple always cost more than a plum? And so you could say in B that yes, we are pretty confident that an apple is always going to cost more than a plum. So
okay.
it's a difficult concept, but remember that you're trying to describe the effect. You're trying to make a conclusion based on the graph. Does your independent variable affect your dependent variable? And if the error bars overlap, then it doesn't. We do not have the data to make that claim that there's an effect.
Mm-hmm.
And then when you're describing your effect, never just say that there's a change. Always say which way that change is. Is it increased or decreased?
And to add on to what Ms. May is saying, and how that effect is affecting the total population. That's the entire population.
Yeah.
I say do you want to switch to talking about controls? Because actually I use this as a lead-in.
Yeah, perfect.
Because students are asking like, well, what's the control? And the challenge with controls is it depends on the structure, the design of your experiment.
Mhm.
So this data is an example where there's not really a control. They're picking 3 fruits that are common to survey. There's no control here. If I can share my screen, I will show
Yeah,
you an example
please.
where there is a continue. Okay, sorry. Where there is a control. This is permission by a student. So there was a student who wanted to know how much fat was in different foods. And she developed her own, she developed her own technique. So she needed a positive control and a negative control in this experiment to verify the accuracy of her technique. And in this situation, she used olive oil because we know that's a fat. And so that's a positive control. If her technique is valid, she will get a result for her olive oil. And she used water as her negative control because you know there's no fat in water. And so she knew with this technique, she should get no result, a value of 0. And so in a lot of experiments, the positive and negative controls act as a verification of your methods.
Mhm.
The positive control should always work. If it doesn't work, then you know there's something wrong with your experimental process or procedure. And the negative control absolutely should not work. Otherwise, that tells you maybe there's contamination, maybe you're doing something wrong. So that's one common use of positive and negative controls, is to give you confidence that your data is accurate and valid.
I also, excuse me, in my class talk about how negative controls can also be used to provide a baseline for certain data. And I use the old school cell respiration lab for this with the peas, where you use beads as your negative control. And if there's any amount of gas consumption at all, then we have to subtract that number from the amount of oxygen consumption we observe in the peas. So that's another important use for a negative control.
Right. Exactly. It can remove background noise. And that gets back to what I think Danielle said about the independent and dependent variables of, by accounting for those other variables, other sources of noise, you're able to be more confident in your claim about the effect of your independent variable on your dependent variable. You're accounting for other possible, again, errors in measurement or background noise. So, and that's the point of any control, is to give you more confidence in your claim that X is really affecting your Y and not, and not being Z. It's not due to Z.
Go ahead.
Oh, what I've been seeing, looking at the 2 samples that were given by the College Board in their videos, and then just other samples of this new format for question 1, is they're often being asked to justify why a particular control was given. And justify is a little bit higher than just describe.
Right.
It's what would you not know if you didn't do the control? Why do you have to do the control? Not what the control is. It's not just something to compare to. It's to make sure that you know that X is actually affecting Y. So, so kind of the describes and the explains and the justifies, those are kind of higher order task verbs. And you gotta bring your game to those a little bit more than just your predictions or calculating or identifying. So those are the ones that are going to take a little bit more in-depth response.
And I would encourage you, use your nouns when you're explaining what the control tells you. Don't just say it tells you that X affects Y, like we're saying here. Use the variables that are in their specific question. Right, so the presence of the peas affects oxygen consumption. Bring it to their question. Use their nouns.
Yeah, never use it. Never use it or they. Just be as specific as you can.
And use complete sentences.
Yes.
Even though you might be able to get away for an identify without
Yeah.
using a complete sentence, the instructions say use complete sentences. So you need to use complete sentences. And it's in your best interest because you're more likely to have a complete thought if you're using a complete sentence. And one thing that I tell my students is it feels like you should write one more sentence that starts with therefore, then write that one more sentence to finish off. And at the reading, they sometimes refer to this as closing the loop. Like the student is almost there, but they haven't quite answered the question yet. So using your therefores and because can be helpful, but avoid writing too much.
Yes.
So
Yeah.
there's this happy balance because your time is going to be short.
Yeah.
You know, please
Yeah.
be cognizant of your time. You have a certain amount of time for number one, a certain amount of time for number two, and then they give you 5 minutes to upload. And I can't stress this enough. When you're doing your prediction and they tell you to justify, please support your prediction with evidence and make sure that you are clear in that. And be, like I said, be mindful of your time.
Yeah. Another thing that can save you time is not restating the question in the response. And I know
Right.
any of you who are taking AP Lang or Lit at the same time, you're going to have to fight your urge to like restate the thesis in the question. But if you can save yourself that first sentence or so of saying, well, one question that can be asked using the experimental data is, that's going to save you some precious seconds that you may need to finish up a response somewhere else.
And remember, you can skip letters and come back to them within a question. So if you answer A and B and you get to C and you're not sure, skip it, go on to D, see if you can answer D. And what I've suggested to my students this year is if they get to a letter and it seems like they have to write a lot, maybe skip ahead, do the other ones and come back to that.
Yeah.
Yeah,
Especially this year's exam. Yeah.
Yep. And it's been stated by the head honcho, Trevor at AP, you can get a good score without answering every section. Rushing through and just slapping something down for 10, for A through H or whatever it is, J, without really putting a lot of thought into your responses is probably not going to get you a very good score. Answering 6 of those prompts out of the 10 and putting some thought and effort and analyzing the data and really thinking about what the response should be is probably going to get you a better score than just kind of rushing through and just throwing some stuff in without thinking.
Yeah.
Don't be afraid to skip. You're allowed to skip
Yep.
and answer them in whichever order you want.
And if you don't finish the 10 points and that clock is running down past the 5 minutes, you need to start working on your submission. Can I say something about submitting?
Oh yeah.
Yes.
So I do the demo. That's without a doubt. Do the demo. Practice submitting every single way with the demo because you never know. You might be set that you're going to type. And then it gets going and you're like, I can't type this. I just want to write it out. Well, now you're writing it out and you've never practiced submitting a photo before. And what if you submit your photo and somehow it's not working? Well, have a backup, know how to put that photo into a PDF, into a Word document, but know what you're uploading. So that demo is really, really important so that you can troubleshoot in that short amount of time that you have. So there's 2 ways to submit. One is a text file and one is a photo file. Don't put a Word document in with a picture into the photo file. It's going to reject your file type. So make sure that you've read through those instructions and practice so that you feel comfortable with that submission. And that's not causing you stress. And don't wait to submit until 30 seconds before that timer runs out.
One other issue I think that some students have had is they've taken a photo and sent maybe with their phone and sent it to their computer to upload it, and then they couldn't find the photo.
Mm-hmm.
So
Yep.
what you want to do is know, where does your computer put the photos it gets? Does it put them in a download file? Does it put them in a photo file? Make sure you know where they're going so you don't have to hunt for them.
Yep.
If you have an iPhone, especially a newer iPhone, your
Yes.
photos do not save as JPEGs unless you changed that already. They save as HEIC files, and those are not accepted by the College Board. So you need to go into your phone and change your settings so that your pictures save as JPEGs or PNG files. I will add to the comments below the video the link to the College Board's troubleshooting site, because there is some information in there about that. So I would highly recommend going and reading that troubleshooting site before the exam even starts, because if there are some preemptive steps you can take to just make things easier for yourself during the exam, that's absolutely worth your time. I also know that the link to the slides below the video right now is not linking to the right slide deck, so I will update that when we're finished as well.
And make sure, you guys, that you sign in 30 minutes before the exam. Do not wait right before the exam to log in, because I know for a lot of tests that have happened prior to this one, which will be on Monday, there's been a lot of fluctuations in, internet. Some people have been logged out. It's just a lot of things that happen. So make sure that you log in 30 minutes before the exam.
And there's things that my son has taken 2 exams so far. There's things you actually have to do before 2 o'clock. You have to type a paragraph. You have to answer some questions. And those all have to be done before, because at 2 o'clock when the exam starts, it doesn't matter that you're not done typing your paragraph. That 25-minute clock has already started. So it doesn't start when you enter. It starts at 2 o'clock. And that's 2 o'clock Eastern time. So make sure you know what time it is for your time zone. I did one of the College Board ones like a student, and I had the timer going, and I was going to upload it to something. And then I couldn't get my windows right on my computer, and I had papers everywhere. My workstation was just a disaster. And so I'm trying to organize, and the clock is going down. My writing time is being taken because I didn't organize myself before I started the exam. So you want to kind of think of that 30 minutes of time, like finding your formula sheet, finding whatever notes you want to have handy. I strongly suggest not having a ton of pieces of paper, because you're not going to have time to look at them, but maybe you have a couple things that are going to help you remember some things. And you got your pages all set up with your APID, and you do all that prep work before 2 o'clock, because you don't want to have to do that during the testing time, because it's just taking time away from when you can show what you know.
Don't forget to submit your answer.
Yes.
Don't forget to submit your answer.
Yes.
Yeah. Don't forget to submit. You do all that work and then you like, okay, I'm done. No, don't forget to submit your answer.
Give yourself the 5 minutes.
Yeah.
Yes.
Yeah.
You're going to get more credit for submitting 6 answers on time than trying to submit answers too late.
Right.
Yeah.
And don't forget to breathe. There's just that whole thing. Take deep breaths. Don't forget to breathe. And just know all this test is to show what you can do in that 45 minutes on Monday. It doesn't indicate what you can do for the rest of your life.
That's right.
It's a chance, it's a check-in, see what happens, and then you go on with the rest of your life and do what you do.
Kids, y'all have done an amazing job thus far. We are dealing with something that is uncontrollable, and whatever you do, wherever you are doing a great job. So don't stress yourself out. You'll be fine.
Yeah.
It's okay.
Yeah. Maybe we can wrap up with just some quick advice for students who are maybe trying to decide what they should be doing to prep for the exam over the next 2 days.
Don't prep on Sunday. Let it go. Yeah. I am a firm believer, and my kids know Ms. Taiski is not playing. When it comes to that Sunday, you need to relax your brain. Today, you do whatever you got to do. Your teachers, I will truly say that AP biology teachers are very, we're very passionate and very protective over content. And, you've been taught very well so far. And, just go to the stuff that you having a little problems with, and just review over that and then just let it go.
Yeah, I would agree with Danielle, with what Danielle just said. If you have to pick something to focus on today, pick the thing that you feel like you need to review the most. Trust that you know enough about everything else, and just focus on those 1 or 2 things where you think you have the biggest room for improvement.
Mm-hmm.
And I would add, get a good night's sleep before Monday and eat some good food, eat some healthy food, whatever is appropriate for the time of day that you're taking the test. Eat some good food before you take this.
No junk food.
Yeah, no junk food.
Yeah, and I would add, looking at a lot of the questions in the chat, there's a lot of focus on small grain size stuff, and worried about memorize or knowing this and knowing this and knowing this and knowing this. Remember that a lot of this exam is what you can do with the data and what you can do with the information. So if you haven't done the 2 College Board practice timed exams, that would be an excellent way to spend some time today, because you'll be practicing questions that have content that require you to use the skills that are the format like you're going to see, and then there's a follow-up with an explanation. And then just get some sleep and have a dance party when it's done. Either together on Zoom with your friends or in your bedroom by yourself.
Yeah, definitely. Yeah. And if you have questions about anything, you can tweet at me. My Twitter handle is Jamie N. Castle. I don't know if anybody else wants to share ways to contact them with last minute questions.
I'm not on Twitter.
Me neither.
I'm at Valerie May. So Valerie May _ May.
Yeah. I'll also keep an eye on comments below the video. So if you have any questions, I'll keep an eye on that over the weekend as well, and try and help anybody out who's got some questions. So good luck, everyone. You are well prepared at this point. You've done a lot of work this year. AP Bio is a really hard class. And so, you should be congratulating yourself before you even take the exam. Mm-hmm. And I wish you luck. I'm sure we all wish you lots of luck on Monday. And thank you for coming today. All right.